So this is Adrift. I love Wendell Berry's book, Life is a Miracle. And in it he says, if we are to protect the world's multitude of places and creatures, then we must know them, not just conceptually, but imaginatively as well. The ocean is my place of contemplation and delight. Puttering in the inter-tidal amidst the scattering of stars and rabbit-eared seaweed, or paddling among the kelp jungles and jellyfish, I am lost in the awe and wonder of discovery. I'm not alone in this. The ocean, our primeval mother, is where life began. A trip to the beach is a sort of homecoming, as people gather there for rest and recreation. I began my love affair with the ocean when I was a teenager. Like many introverted teenagers, I was full of angst about the future, and spending time at the beach was a way of decompressing, a way of calming down and gaining some perspective. It was kind of like a reset button. You know, I could go there and just get a clean slate on everything. And I think that this affinity with the ocean is a common experience for people, and I think it's the reason why the seaside is such a popular place for vacations and recreation, or more aptly, recreation. We really do go there as kind of a reset. And my affinity with the ocean led me to study marine biology and aquatic ecology here at UBC many years ago as a young person on my first time around the university circuit. And it's been a huge influence on my art practice as well. Much of my artwork over the years has focused on life in the oceans. So this might seem a little bit disjointed, but this seemed like a way to tackle this talk today. So all the text that's on the screen is out of the artist statement. So with climate change, the oceans are becoming warmer and more acidic, less hospitable to life. I think our awareness of the problems of atmospheric carbon loading began in the 1950s, and we started to become aware of the possible ecological impact that would have. And over the last 60 years or so, it's become clear that this is having a fairly significant and critical impact on plankton ecosystems as well. A study published in Nature in 2010 reported that marine phytoplankton, which are responsible for half the carbon capture and half the oxygen production in the world, and are the foundation of global food chains, they have declined by 40% over the since 1950. So this is it's kind of a scary thing. You know, as we're pumping more oxygen or more carbon dioxide into the atmosphere, the oceans are becoming less able to cope with that. Warmer surface water temperatures have led to increased seawater stratification, which has meant a decrease of nutrient cycling in the ocean, so there's less micronutrients that plankton need to grow. And phytoplankton are just simply unable to keep up with all the carbon dioxide that is being dissolved into the oceans, and so it's sitting there in the form of carbonic acid and creating ocean acidification. And this is interrupting critical life processes. So in a study, a research group from Princeton University in 2011 published reported that ocean acidification decreases calcium, the saturation of calcium carbonate in the water, which makes it harder for organisms that are building shells to do it, because the shells the calcium carbonate just dissolves in the water. So we're talking about all the mollusks, the corals, phytoplankton such as foraminifera and coccolithophores. These are all organisms that create little shells, and when that happens, when they're no longer able to create their shells, they are no longer able to live. And this doesn't just have an impact on the benthic things like clams and snails and stuff. It also has an impact on other organisms. For example, nudibranchs are organisms that don't have shells in their adult form, but they're part of the plankton community as in their larval stages, and they actually do form a shell at that stage. So they're particularly delicate and vulnerable. The group from Princeton also reported that decrease in pH changes the bioavailability of nutrients, essential algal nutrients like zinc and iron, and that increases in carbon dioxide affect the ability of photosynthetic organisms to synthesize, and may also change the composition, the carbon to nitrogen ratio. And nobody has any idea of what the impact, the long-term impact of that's going to be. Forgot to change the slide. Could have been looking at this instead. Anyways, optimists have reasoned that life on Earth has survived catastrophic change before and will again. But what kind of impoverished life awaits those who survive? How have we come to this impasse in our relationship with nature? And what kind of shift in thinking is required to turn things around? Over the past decade, a number of writers have endeavored to deconstruct what we mean by nature. William Cronin, in his essay, The Trouble with Wilderness, has suggested that the trouble with nature or wilderness is that it is largely an illusion of a distant and undisturbed refuge, and its existence permits us to abrogate our responsibility to look after the places where we actually live. Cronin says that we've got this romantic vision of wilderness as some kind of sublime, pristine reserve that's apart from us, when in reality, as humans, we're part of nature. We set aside these wilderness preserves almost as an insurance policy in case of ecological catastrophe. And we have this notion that as long as we have these undisturbed areas to fall back on, if all ecological hell breaks loose, we can develop the rest of the landscape to suit our growing human needs. And it's kind of an absurdity. I don't know how many of you, probably all of you, have been up in an airplane. I'm always shocked when I get a window seat and I look down on the landscape and I see how little of it has been left untouched by humans, how much of it is covered in concrete or has been claimed by agriculture. And this has been my experience all over the world, not just here in Canada. Cronin goes on to argue that since humans are part of nature, the only way that we can save ourselves from ecological catastrophe is by living in a more ecologically responsible way. Two other philosophers, Bruno Latour and Timothy Morton, believe that the problem is that nature, as a monolithic and immutable source of raw materials and a sublime backdrop to human activity, simply does not exist. What does exist is a diverse collective of plants and animals, earth, air, and water. Latour and Morton insist that we need to consider the interests of other species and recognize that their personhood and right to exist. Since our vulnerabilities are interwoven, our survival is contingent on theirs. Latour has proposed a new way of including ecology within politics, that is, a new constitution where all stakeholders, human and non-human alike, are given a voice for the common good. He suggests that scientists, in their capacity for inquiry into the non-human world, speak on behalf of the non-human. Environmentalists have long argued that what's needed is a more comprehensive accounting, a complete global economy, one which counts everything and everybody, which includes all living things, not just money. We need to recognize that the natural resources required for life to go on go beyond just oil and gas and mineral reserves. That every part of the ecosystem has integral value and is critical to our survival, whether we're aware of what that value is or not. But can scientists alone adequately do this? Personhood implies particularity. Wendell Berry has noted that while scientific inquiry focuses on the specific, its interpretation and application is generalized, sometimes to the point of doing violence to the particular. Art, on the other hand, focuses on the particular as subject. The role of the artist, to paraphrase Paul Klee, is to make visible the invisible. The role of the scientist is to make known the unknown, and perhaps both can speak on behalf of the non-human. I would argue that while science can tell us what the problems are, the impediments to solving our problems are largely cultural and political. And we have this really strange relationship with science. On the one hand, for many people, science has assumed kind of a metaphysical status, becoming, in the words of Wendell Berry, modern superstition. We are eager to accept science's dispensation of technological providence and promise, and I think that many people sincerely believe that somehow science will get us out of this mess, the mess we're in, and that some brilliant scientists will come up with a solution. But on the other hand, when science tells us that we have a problem, we're very reluctant to believe it. And we are simply unable to conceive of a world that is very different than the one we wake up to every morning. Slavoj Žižek, in his book In Defense of Lost Causes, refers to Donald Rumsfeld's famous elucidation on how we have the known knowns, the known unknowns, and the unknown unknowns, and he completes this quadrant then by saying we also have the unknown knowns, things that we know, but we are not aware that we know. And he's talking about these archetypes in our subconscious. It's these invisible unknown knowns that resonate within us when we look at art or listen to music and poetry. These unknown knowns that address our profoundest longings and for meaning and connection, and inspire us to heroicism even when it goes against our own self-interest and convenience. And it's this deep intrinsic knowledge to which art speaks. And that's why I think that art is important in trying to address some of these issues that seem more scientific. For some time, I have contemplated how to merge the worlds of art and science in my own practice. I've been inspired by Johann Wolfgang von Goethe, the 18th century poet and playwright. That's Goethe. Goethe was a German writer and politician, but he had he was kind of an amateur scientist and was really interested in plant morphology and color theory and wrote a lot about these things. His approach to science was to focus entirely on observation. For him, the phenomena embodied the theory. And the other inspiration was Ernst Haeckel, who was a German biologist, naturalist, and illustrator. He discovered and described and named thousands of new species and created exquisite illustrations of approximately 4,000 different marine plankton. And these are diatoms, which don't actually look like this. I think he was from that Victorian age when they like to elaborate a lot. So these are Victorian diatoms, not real ones. So I took my cue from them. For 1 year, I employed methods from science to collect and analyze monthly plankton samples at Whaler Bay, BC, and then documented and interpreted my observations through images. I wanted to say here that it's been a good 30 years since I was actually in a real lab working as a scientist, and I don't consider myself to be a scientist anymore. I pretty much just a rank amateur nowadays. And the so-called scientific work that I've done here is pretty crude, without any of the rigor that real science requires. You know, I did not have access to an oceanographical vessel with downriggers and all that gear and special scientific collecting equipment, so I had to make do with homemade stuff. So I built my own plankton net with silkscreen mesh, which is actually really great because it's specially woven so that the little spaces don't, the mesh size doesn't change as you force water through it. And I used a plastic embroidery hoop for the frame and got a little plastic jar, cut a window out of it, glued mesh on it. So it screws onto the end really easily. Great for collecting. I had actually borrowed a net from the University of Alberta and found it a lot more cumbersome than this one, so I was quite happy with my construction. And to check water turbidity, I made my own Secchi disc. The Secchi disc is basically a big metal disc that's got very strong black and white markings on it, and basically how you measure turbidity is you drop the, it's attached to a rope that's got markings on it for the depth, and you lower it down into the water until you can't see anymore, and that depth is the Secchi depth, and it gives you an indication of the water turbidity, which in turn, unless there's been a big rainstorm and you're near the coast and there's a lot of gunge in the water, gives you a good idea of how much plankton is in the water, particularly phytoplankton, because it's largely the chlorophyll that absorbs the light and creates the turbidity. Anyways, I got an old saw blade and covered it with black and white duct tape, and that worked really well. And I used socks filled with rocks for weights. Also works really well. And we did this at Whaler Bay because we have a little cottage near there, and Henry drove me around, Henry, my husband, and lab assistant, field assistant, drove me around in the boat. We had 150 meters of nylon line that we would drag at about 30 degrees, so for a maximum depth of about 10 meters. Over 2 minutes, then I would haul it all in, and so we get this continuous tow from 10 to 0 meters, and over a considerable distance usually. So that, I thought it was, for my own experience collecting plankton when I was back in grad school, it seemed like a reasonable way of doing it. And I took monthly samples, which really aren't adequate to get a comprehensive idea of what's going on in the water. But I was living in Alberta at the time, and the Alberta Foundation of the Arts would only give me enough money for airfare to come home once a month. So that's how it, kind of the experimental design was largely dependent on logistics. So probably missed some blooms, particularly in the summer. You really kind of need to be sampling every week to really get a good idea of what's going on. But the purpose of doing the study in the first place, was to have a look to see what was out there. You know, we had this place for ages, and I had no idea of what kind of plankton was out in the water. And I wanted to see what kind of changes happened during the course of the year as well. So that's kind of what this study was about. And it was about seeing what kind of weird and wonderful things there are out there in the water. Initially, when I started collecting samples, or when I was about to start, I thought that I would preserve the samples and then look at them in the lab under a microscope and count them the way I had done 30 years ago. And I thought it would be very cool, since I was connected to this large university, to do electron micrographs, because I think they look really great. But when I looked into this, I realized that I just simply wasn't equipped to deal with the incredibly toxic chemicals that are required to do that. And so I decided rather than to collect samples, I would photograph the live samples. So I got an attachment to attach my camera body to the microscope and took pictures of the live organisms. And it turned out that there were a lot of advantages to doing it this way. One was that I got the true colors, the chemicals that preserve the organisms really bleach them. They make bits fall off of them. Particularly, electron micrographs were pretty nasty. The chemicals involved in preserving the organisms for that process are really nasty and do make little bits fall off. So I thought that was great. The other thing was that I wasn't coping with mounds of jars of samples to store. And the last thing was that I could take videos of these live organisms, which was something I hadn't counted on. Another thing that I was, so this is kind of a typical sample under the microscope. This is what they look like, just kind of random bits and bobs depending on what was in the water. And the other thing that I was really interested in was figuring out a way to present the data in a way that was a bit more accessible to people. I think that scientific journal articles aren't particularly accessible to the non-initiated. And I wanted to see if I could find a more inclusive, meaningful, and engaging way to express what I found. So what I did was I counted the numbers of different organisms. I made up my own catalog that I didn't really worry about the scientific names of things. I kind of fell back on Goethe and just worried about the phenomenology, the observation. If it looked the same, I assumed that it was the same. So I created a catalog of probably about 200 different organisms. And then I just counted how many there were of each. Usually enough different samples until I got about 1,000 cells, or 1,000 organisms. And from that I calculated then the proportion of each organism in terms of numbers in that whole sample. And I gave each organism a color. So the bar on top kind of tells you what the distribution is. So it gives you an idea. These round guys, diatoms, were the most plentiful in the sample, and so that's the biggest bar. And the size of the bar relates to how many of each organism. And when you look at a whole bunch of them, then you start to see the changes that happen. Some months there was a predominance of one species over others, and in other months, for example, the second one from the left, it's a bit more evenly represented. These images were constructed from the samples, from the photographs, because obviously I wasn't going to find an image that was perfectly representative of the numbers that I got, so I constructed them and used the Secchi depths as a way of dealing with the density, the overall densities. So in looking at this, you could see that there's fewer cells in January than in April. I think it's the April sample that's in the middle there. But anyways, so I hope that gives you some idea of what those things are about. So when you go into the gallery, you'll see them month by month, and you can see the monthly changes, and you can see some species popping up and others dying out each month. I was also really interested in the forms of these organisms, particularly the animals, because some of them are really pretty weird and wonderful. And I'll pick up on that in a minute. Images of nature in art have historically been relegated to the fringes as naturalist illustration and pastoral or sublime landscape. I elected to work with portraiture, which is an art form that has been traditionally reserved for humans. And I made portraits of individual organisms, carefully avoiding the scientific habit of splaying them out for identification, but working with them as characters in the actual poses I observed to convey their ecological importance. I made the portraits human life-size. While the images are mimetic, they reveal forms that are unfamiliar and fantastic, making visible the invisible. I chose the medium of printmaking, which I think is fitting for representing living things. The art of making images from a plate or stencil imitates the reproduction of life, each living organism an expression of a genetic matrix. Printmaking embraces many forms, from the ancient traditional craft of woodcut to the highly technological media of digital photography and video. Woodcut, where every mark is made by hand, embodies the subjectivity and interpretation of the artist. There is an element of the contemplative in the quiet deliberations of where and how each mark is made on the surface of the plate, requiring patience and devotion reminiscent of monks illuminating medieval manuscripts. So all of these critters, the big critters on the walls, were done as woodcuts. So I made these large zooplankton portraits using woodcut, and the plates were made from masonite and carved using a Dremel tool. Because of the annoying noise of the Dremel tool, I was relegated to a wood shop in an ancient maintenance building in the basement of this. No windows, hardly any ventilation. I spent many, many hours in this dungeon with my earphones and iPod, earbuds in there, hours and hours and hours, months and months. And I felt quite isolated. I felt quite a bit like a monk, actually, down there. Anyways, so that's how these images were done, one at a time. Translating a map that I made from the photographs into kind of a topographical map first. That was transferred onto the plate, and then each area of different darkness was translated into marks. And photography and video, mediated by technology and detached from the artist, is the medium of choice for depicting reality. Like science, it presents the illusion of objectivity. Using digital media, artists combine diverse images into cohesive compositions, much as scientists employing statistics transform apparently random data points into coherent narratives. So in these images, I used both the digital photographs as well as the woodcuts and combined them. And I was trying to convey these aspects of observation in the digital image, printed on paper, mounted on the board, and interpretation in the woodcut that's printed on silk and suspended over the digital image. In the larger, so obviously there's less detail and more imagination in the woodcut than there is in the photograph. So that was kind of the point of those. And then in the larger images, where I combined the digital with the woodcut, the digital images are fragmented, much as scientific information is fragmented. Scientists can't look at everything, so they choose, disparate places to examine and then try to piece together knowledge from this patchwork of information. I think we sometimes forget that science, like art, is a human construct, and that it is also subject to human frailty, bias, and subjectivity. And this isn't meant to dismiss science, not meant to be disrespectful, but simply to acknowledge that there are flaws. And the more that scientists are able to recognize these, the more they are able to be objective and to be better scientists. And I think it's also important to acknowledge in the big picture of things that science simply cannot fix everything, especially in our current climate. We're almost done. The dichotomies of science and art are mirrored by those of observation and interpretation. Of new technology and traditional craft, of objectivity and subjectivity, and ultimately of object and subject. In trying to bridge this divide, I often find myself adrift in a tide of contradiction. The liminal space between the two is fraught with collision. They crash against each other like the sea against the coastline. The water is murky and difficult to navigate. The turbulence regenerates nutrients that stimulate growth. Here, dichotomies can meet and be reconciled. The old and new can work together as observation and interpretation merge. The moving picture, fluid and ephemeral, can present a healing dialectic where observer becomes subject and creature becomes character. This last video is from a part of the exhibit that I wasn't able to put up here at the BD because of space restrictions. This video was displayed. We had a small white room that was 14 by 14 feet in diameter, 12-foot ceiling. It was all white inside. And in the center of the room there was a 3-foot diameter mirrored half a globe. And the video was projected from the ceiling onto the globe. And so this video then ended up going on the floor, up the sides of the wall, across the ceiling, down, and back under the globe. And it just, the jellyfish, we were basically surrounded by jellyfish in the room. The reason I included jellyfish is because they're part of the plankton world as well, but they're not invisible for the most part, except for when they're real tiny babies. But they're also kind of scary because they are sort of the cockroaches of the sea. You know, as everything else collapses, jellyfish become more and more plentiful. And yeah, anyways. So I am still struggling with this whole idea of trying to find common ground between art and science. I'm not really sure that is possible, but I'm hopeful that it is territory that can teach us a lot, that it is fruitful ground for exploring ideas. So far in this pursuit of it, I have learned a few things: that both disciplines require a great deal of creativity and ingenuity, that both depend on our skill and our honesty, and that both are integral pieces of our humanity. That's it. Thank you very much for listening.